welcome to the Engineering Biology Research Consortium Education module on synthetic biology and machine learning. I'm Mary Dunlop, a professor of biomedical engineering at Boston University, and in this series, we're talking with researchers who work at the interface of synthetic biology and machine learning about projects in their groups and ideas for how to learn more about this area. So we're joined today by Jonathan Stokes, a professor at McMaster University in the Department of Biochemistry and Biomedical Sciences. Welcome, Jonathan.
Hello.
So in this discussion, we're going to be talking about a paper that he published on using a deep learning model to predict antibiotic activity based on chemical structure. And the study identifies a new antibiotic called halicin, which is structurally distinct from many known antibiotics and shows broad spectrum efficacy against bacterial infections when it was tested in mice. Jonathan was the lead author on the study, which he conducted when he was a postdoctoral scholar in Jim Collins's lab, and it was published in the journal Cell in 2020. We'll get into the machine learning parts of this work in a little bit, but can you start out by just telling us about the overall goals of the study?
Yeah, so I'll tell you about, kind of how I approached this paper going into it. So my background is in antimicrobial chemical biology, and I'm interested in discovering new antibiotics to overcome resistance, right? So, the whole rationale behind this project came from a largely historical perspective. And that's what I'm showing here. So this is just a timeline of our antibiotic discovery, right? We discovered our first antibiotics in the late 1920s. But what we can see, right, is that our productivity in antibiotic discovery was not, necessarily consistent over the past 100 years, right? We can see that, the majority of our unique structural and functional classes of antibiotics were discovered between the 1940s and the 1960s. Right? This is the so-called golden era of antibiotic discovery. And it's a period of time characterized by high productivity in discovering different structural and functional classes of antibiotics, right? And this was done largely through screening secondary metabolites produced by soil-dwelling microbes. But you can see, right, come the middle of the 1960s, our ability to discover a whole ton of different antibiotics decreased, right? And we ran into something called the dereplication problem. And what that is what happened is we kept discovering the same antibiotics over and over and over again.
Yeah.
So in more recent decades, right, starting kind of in the mid 1980s, perhaps, we turned to small molecule screening in an attempt to find new antibiotics. And what this looks like is chemists would build these giant synthetic chemical libraries, and then we would screen them for, some biological property of interest, whether it was an enzyme inhibitor or something that inhibited the growth of bacteria in a dish, whatever. Mostly the former. But, through, however many decades, 3 decades more of screening synthetic chemical libraries for new antibiotics, we haven't discovered any that are used in the clinic. So we find ourselves in this so-called discovery void, right? Where we haven't discovered any fundamentally new antibiotics in decades. What's interesting, like this problem in and of itself would not necessarily be a problem, right? We have a bunch of different antibiotics and they all work and whatever. But what accompanies this lean antibiotic pipeline, right, is the global dissemination of antibiotic resistance determinants, right? So we're getting to a point where a lot of our once good antibiotics don't work anymore, right? So we need to discover new antibiotics, rapidly and ideally inexpensively, right?
Yeah.
And I felt that the application of machine learning and deep learning would afford us the opportunity to discover such molecules rapidly and inexpensively, right? So that's where this whole project kind of came from.
Why is this particular task well suited for machine learning? Like, what kinds of data do you have, and how much of it is there?
Yeah, so it's a really good question. So let's look at this. So I'm going to describe this in kind of 2 parts. All right, so let's say we have some chemical landscape, right? So here we're looking at a giant collection of chemicals, 10 to the 8 molecules, right? So let's say we wanted to try an old-fashioned, like conventional small molecule screening approach, right? That's what we're looking at here. So I go assemble a chemical screening library, and if I'm a really large pharmaceutical company, let's say, I can build a library of a couple million molecules, right? So I'll build this library of a couple million molecules. I'll screen it for, some antibacterial property, and I'll get my hits. It's a low hit rate. It's generally far lower than 0.5%. And then I find the molecules that I'm interested in, and then I figure out what they do and see if they work in a mouse, et cetera, right? But this, what I'll call a conventional screening approach, is suffers from, I would say, three drawbacks. The first one is it's really expensive, right? Because if I want to screen 2 million molecules, I have to buy them, right? It's time consuming because I have to test all of these molecules individually in the laboratory. And three, and I would call this perhaps the most significant problem, is that the chemical space that's explored in some of these screening, like high throughput screening collections, is not as broad as we would like, right?
Yeah. Mhm.
But we can contrast that with, a machine learning guided approach, let's say, where we can train a model on, thousands to maybe tens of thousands of molecules, right? And once we have that model trained on, thousands to tens of thousands, that's a very small screen to perform in the lab. It's easy. Once we have our model, we can then take that model and run predictions on vast chemical spaces in silico, right? We can run predictions on hundreds of millions to billions of molecules, right? And
Right,
we can
right.
do this orders of magnitude faster than we can empirically test molecules in the laboratory, right? And this allows us the opportunity to explore huge chemical spaces that we never could in the lab, right?
Right.
And it's by being able to rapidly explore these vast chemical spaces that improves the probability that we're going to discover molecules that have properties that we want, right?
Yes.
So
Uh-huh.
I come from an experimental background, so I always focus on, okay, what does the actual data on which we train these models look like, right? And I'm obsessed with data quality. So this is what we ended up training this molecular property prediction model to predict new molecules against E. coli, right? So in the laboratory, so we curated this data in the lab. This is real data. These are 2,500, a bit more, small molecules. These are all very structurally diverse. And what we're looking at is the growth inhibition of E. coli by each of these 2,500 molecules, right? So on the vertical axis, we have growth. And then this is just molecule 1 to 2,500, right? And what we wanted to do is classify this data as either growth inhibitory or not growth inhibitory, right? That's pretty straightforward. So we took all the chemicals that we deemed did not inhibit the growth of E. coli, right? Colored them blue, got all the structures, and then just labeled them as 0, right? And then we took all the molecules that we said these inhibit the growth of E. coli very well. We got their structures, then we labeled it 1, right? So this was the input data to our model.
Can I ask a little bit about the like this binary classification scheme that you use, where it's either 0 or 1?
Yeah,
You could have done other things like looked at the relative growth on a scale of 0 to 1 in some sort of continuous fashion. Like, why did you end up deciding to do the binary classification?
yeah. So we had two options, right? We either could do a binary classifier or a regression model. And a regression model would be one that takes into account all of the like little differences in growth between
Yeah.
0 and 1, right? We actually tried to do a regression model at first, but we didn't have sufficient amounts of training data with which to make a robust regressor.
Oh,
So
interesting.
we turned it into a binary classifier, which we observed worked a lot better. But it's interesting that you bring that up because I like the idea of having a model that can predict how potent a growth inhibitor something is, right? As opposed to it will be or won't be. So that this is one of the things that we're working on now is building um, regressors that can predict the potency of molecules against some pathogen of interest. It just the issue is we need to acquire, like at least an order of magnitude more training data than we have here. So once we had our right, our structure property pairs, that's what we're looking at here, molecular structure property. Um, we train our model, which we will touch on in a little bit, right? And then that afforded us the opportunity, once we had our model all trained up on the 2,500 structure property pairs from our training data set, we could then show the model any number of structurally diverse chemicals. And then based on what the model had learned during training, it would output a number between 0 and 1, right? And as you would assume, if the output number was closer to 0, the probability of that molecule being an antibiotic according to the model was low. And if it was approaching 1, the model was pretty sure that it would be growth inhibitory in E. coli, right?
Right, right. Yeah, so although you're training it on sort of zeros and ones as possible outputs, it's actually predicting a probability that's either 0 or 1. So you could get a number in between there.
Yeah, and that's just, based on what the model learned during training, the probability that that compound has the activity of interest, right?
Yeah. Can you talk a little bit more about how you represent these chemical structures numerically?
Yes, so that's a really important question, right? So this is the approach that we use is called a message passing neural network, right? And the problem, it's not a problem. The challenge is, right? So if we have a structure of a molecule, right, and we have some property associated with it, we need to encode this structure in a way that we can train a model with it, right? We can't, we have to encode this in a way that makes sense. So there are a whole bunch of different ways um, that folks have attempted to encode molecules as vectors, right? This goes back to like the invention of quantitative structure activity relationship studies. This was like back in like the 50s, right? Where people would take molecules, turn them into vector representations, and these vector representations might encode things like the molecular weight and the hydrophobicity or like the polar surface area. These were like computable molecular features.
Okay, but
And
you have to know something about like which
yeah,
properties are interested in
e-
hand
e-
code that in. Yeah.
exactly. So those rely upon like expert knowledge. Like it's like human intervention to define what the vectors would be, and therefore humans have to know what the important molecular features are. But the question becomes, what if we don't know necessarily what these important molecular features are? How can we develop a model that can learn that automatically, right? And that's the power of some of these more contemporary deep learning approaches, right? So let's use this molecule. Let's say this was a molecule from our training data set, whatever, right? What we want to do is we want to take this molecule and encode it in a way that we can, train a model. So one way we can turn this molecule, the structure of this molecule, into, a molecular representation is by, in essence, teaching all of the parts of the molecule about all of the other parts of that molecule, right? We need to teach the chemical about itself. So what does that look like in practice? So this is a really simplified version of what we did, but it captures all the important bits. So we're looking at this bond and it's 2-1, right? We'll call this bond 2-1. And it's represented here by this three-dimensional vector, right? And this could be, it's a single bond and it's stuck to 2 carbons and it's not aromatic, right?
Okay.
Whatever. So here's that bond 2-1. What we want to do is we want to teach bond 2-1 about itself in the context of its local molecular neighborhood, right? So we're going to look at all the bonds that feed into it, right? So going from left to right across the molecule, we can see that this bond 3-2 feeds into 2-1, and this bond here 4-2 that also feeds into 2-1, right? Now, as one would expect, so bond 3-2 is represented by a three-dimensional vector, and so is bond 4-2, right? So in practice, what we do is we take the vector that represents 3-2 and the vector that represents 4-2, and we just add them together, right?
Okay.
So then we take this summed vector, that's a sum of 3-2 and 4-2, and then we concatenate it with the original vector for bond 2-1, right? So we've just taken the vector for 2-1 and then this summed vector and just stuck them together. Then,
Uh,
Oh, sorry.
why does the summation element of this? What's the logic behind why you would sum 3-2 and 4-2?
So that was purely based on empirical. This happened to work the best. I don't think you have to necessarily like sum them. You could do some other operation, but summing worked fine, right?
Okay.
So then you have this vector. You know, right now it's a six-dimensional concatenated vector. This goes through a feed-forward layer. So this is itself a feed-forward neural network. And then what you end up with is a new vector representation for bond 2-1. And this is an updated bond vector because it contains information about 2-1 as well as 3-2 and 4-2, right?
Okay. Okay. And is this the where the message passing name gets comes from, where it's like it's passing information from? Okay.
Exactly. So through this operation, we've passed messages, we've passed information from 3-2 and 4-2 into 2-1, right?
Okay.
So you can imagine if you do this message passing operation repeatedly, eventually these messages are going to make themselves across the entire molecule, right? And you're going to be able to have a vector representation that captures the molecular complexity of that molecule. And it's this vector representation that captures the entire compound that is then used for training and is then updated during training, right? To result in a molecular representation that is optimized for the specific property prediction task at hand.
Just to ask a few more details about this one. So the feed forward network that you're describing in the where you go from the concatenated vector to the new bond vector,
Mm-hmm.
how do you know what the like weights for the parameters for that network are? How do you decide those?
So all of those weights are updated during training, right? And this becomes one of the questions of, okay, how do we know our model is working, right? And that brings us into this, you know, interesting set of questions like, how do we know whether the model is sufficiently trained or whether it's overtrained, and like, how do we know
Right.
that this is going to work, right? So, um, all right. So let's just say let's not talk chemistry for a sec. Let's just talk like general, classification of like two things, right? So here we're trying to classify stars and circles, right? So we have our circle set and our star set, and what we want to do is train a model that captures, the the inherent structure of the data, right? So we can see, like, let's look at this plot. So we have, two classes of data. We want to separate them in a way that makes sense, and we can see just kind of like by eyeballing it that this art kind of makes sense, right? It works well. It can more or less most of the time classify stars, and it can more or less most of the time classify circles, right? So you know there are then if this is like I call this is like the Goldilocks zone, right? Like we have a model that has learned enough about the inherent structure of the data in order to make robust classifications, but it's not underfit or overfit. Underfit would be the case where we insufficiently train our model, right? And it fails to capture the inherent structure of the data set. So it doesn't really do very well when it comes to any classification tasks, right?
Yeah, like
Conversely,
sufficiently,
though, oh, sorry, continue.
or it looks like in this case, and you're just using an inappropriate model here to like maybe a linear fit between them isn't sufficient to capture the complexity.
Exactly. Like, so this would be like a simple linear classifier, and it is too simple to sufficiently capture the complexity of the data, right? Now, if we go to the other extreme, let's say we way overtrain our model, and now this model is what we would call overfit to the training data. And so we can see, right? So if we overtrain the model and it's overfit, we can, during training, right, we can classify stars from circles 100% of the time. But this model has learned a lot about the noise of the data set, right? So the problem with this is, let's say now we have a new data point and we're going to tell this model, okay, make a prediction of this new data point. the probability that it's going to make a correct prediction is very low because it's learned too much noise, right? So models like this are not able to generalize very well, right? And let's now take it back to the problem that we were trying to solve, right? We want a molecular property predictor to be able to predict structurally novel antibiotics, right? So if we had a model that was way too overfit to our training data, when we showed that model a structure of a chemical that it had never seen before, it wouldn't be able to make a very robust prediction because it was overfit to what it learned during training, right? So that's why we want to avoid overfitting. And of course, underfitting, because underfitting just means your model didn't learn anything important from your training data at all, right? So it's like, I always think about it, it's like Goldilocks, right? It's like this one's too cold, this one's too hot, this one's just right. So this is the way I kind of think about it, right? And then, there are a bunch of ways that, investigators can go about avoiding overfitting, for example, because that's a big problem. So here's one. All right, so let's say we have 1,000 molecules, right? 1,000 molecules that we gathered data for, and let's say it was looking at growth inhibition of E. coli, right? So this is just a training data set of 1,000 molecules. Well, what we can do, let's say we take that 1,000 molecule training data set and we break that up into 5 chunks, right? So each chunk here has 200 molecules out of the 500, right? What we want to do is train a model and then try to get a good understanding of how well it is fit to the data and how well it is able to generalize, right? So what we're going to do is we're going to take 200 molecules, so one chunk of this data, and we're going to put it to the side, right? And we're going to call that our holdout set. We're not going to show that to the model during training, but what we're going to show to the model during training are these 4 chunks of 200. So it's going to learn on 800. And then what we'll say is, okay, once the model has trained whatever specific model we're interested in, we're then going to run predictions on this holdout set and see how well it did, right? How well did it classify hits and non-hits from this holdout set based on what it learned during training from these 4 sets, right? That makes sense. And then what we can do, though, is we have to ask a question. Well, what if this 200 molecules wasn't the actual best set to use as a holdout set, right? What if there was important stuff in here that we wanted our model to learn? Well, we can avoid that problem by just running this experiment multiple times, changing the holdout set every time. This is a very common approach. It's called k-fold cross-validation. Here, it's k equals 5. So it's 1, 2, 3, 4, 5. So this would be 5-fold cross-validation, right? So what we do at the end, so these are 5 independent experiments. And every time we get some prediction output, or some performance output, rather, right? How well did the model trained on these 4 chunks classify the molecules in this 5th chunk, right? And then what you do is you just average the performance across all of these 5 folds. And then we say that's a good estimate of how well our model performs, right? Whether we're overfit or underfit, or whether it's just right, right? And then you can go in and try to, either change the type of model that you're using or optimize your current architecture, whatever you want to do, right? And that gets to this. So you can see high train error, high holdout. What does this all mean? Well, an underfit model, right, makes a lot of errors during training and obviously makes a lot of errors on your holdout set because this one didn't learn anything, right? An overfit model makes no errors during training, right? Because it's learned everything there is to learn, but there's a really high holdout error because it can't generalize very well, right? Whereas, your Goldilocks model has a low training error and a low holdout error. But the other way that, makes me happy as an experimentalist is you just gather more training data, right? So something that I like to do, and we're lucky, we're lucky because, we do mostly wet lab stuff. But what we'll do is we'll train a model, doing this type of stuff. Then we'll actually run predictions on some in silico data set of chemicals, right? Then we'll curate those molecules and test them in the laboratory, right? And then we can then take those additional molecules, we can say, then we can retrain the model. We can tell the model, hey, you were wrong, you know, this percent of the time with these molecules. You were right this percent of the time with these molecules. Here's, here you can retrain yourself, and now you won't make those mistakes again, right? So you can refine your model with iterative cycles of running predictions and then empirically validating the predictions, retraining, right? So that's how it's a delicate balance of, in silico validation to avoid overfitting, and then going back into the laboratory, gathering additional training data in the appropriate chemical space, right? And then you can make your model as robust as you can for some specific chemical space in which you're particularly interested.
Would never want to train on something that you then are using to evaluate like sort of final performance of it. Yeah.
Oh no, that's a super important point, right? Like, yeah, you're never gonna train on molecules that you then, test on, because it's cheating, right? It's like getting all the answers to a test, right? because then because then you'd think, if let's say we did that. Let's do the experiment, right? We're gonna take a model, and we're gonna train it on, or like, we're gonna take, let's say we don't have a holdout set, we're gonna train it on everything, right? And then let's take a subset of that everything, and then you run predictions on it, right? Well, you are like, the model already knows the answers. You're going to have a really high prediction performance, right? It's going to work really, really well. But now let's say you take that model, and then you go and run predictions on a set of chemicals it has never seen before, right? It's going to be making a ton of wrong answers, because what you observed, the performance that you observed by running predictions on molecules it had already trained on, is not indicative of the real world performance. So the whole importance of ensuring that our model is what I'll call it the Goldilocks zone, right? It all relates to this problem. So what we're looking at here, so everything in blue is a molecule that was used during training. Everything in red is a molecule that was used as our prediction set, right? So the red molecules are compounds that we don't know what they do, and we want to see which ones are antibacterial. And what we're capturing here, so this is just a plot of molecules in two-dimensional chemical space. So molecules that are, or dots that are really far apart, are structurally very different, and if they're close together, they're structural analogs, right? So the importance of having a model that's trained in that Goldilocks zone is the ability to generalize, right? We want our model to understand, or have a reasonably good understanding of chemical space, from the very left side of this plot to the very right. We want it to be able to generalize and make accurate predictions very well, right? And you can imagine, if you had a model that was overfit, it would work really, really well against a whole bunch of analogs. But as soon as you had to make it go farther out in chemical space, it would fail, right? And that's the importance in molecular property prediction of appropriately training your model.
Although we're focusing on the deep learning modeling parts of the work, the full paper has some really beautiful results on identifying new antibiotics. I'd be curious, can you give us a quick summary of these findings? And I would just encourage people who are interested in knowing more to check out the full paper, because we're really just touching on a small part of it here, but it's fantastic.
Yeah, so I'm just going to summarize really, really quickly some of the, primary in vitro results that we observed. So when we trained our model, we ran predictions on an in silico chemical data set, and we came across this molecule here up in the right corner. We named it halicin. Um, we selected this molecule for a number of reasons. It was predicted by our model to be antibacterial. And then the other important feature of this molecule is it was structurally divergent from known antibiotics. So during training, like in our training data set, we had more or less the universe of known antibiotic drugs, right? And so one of the criteria that we use to prioritize chemicals post prediction, right? How do we, we have a bunch of predictions. How do we know which ones are interesting and the ones we want to follow up on in the lab? One of the criteria was that the structures of the molecules had to be different from known antibiotics. And this one fit the bill. It was both strongly predicted to be antibacterial and structurally different. So we tested it in the laboratory. So what we're looking at here is growth of E. coli as a function of the concentration of halicin. And we can see that it took 2 micrograms per milliliter of halicin to inhibit the growth of E. coli. right? So that's potent, right? That's on par with known antibiotics. And the next question that we had, right, to get into a little bit more biology is, was this molecule bactericidal or bacteriostatic? Was it bactericidal? Did it kill bacteria? Or was it bacteriostatic? Did it just inhibit their growth, right? So we can do an experiment where we monitor the cell viability after treatment with halicin. So what we're looking at on the vertical axis is viability, right? And again, this is halicin concentration. And then each of these different colored lines is a different duration of incubation with the molecule, right? And we can see that with increasing durations of incubation, we lose viability. Therefore, this molecule halicin is bactericidal, right? Once we understood this, we had a very obvious next question, right? And the question was, well, the structure of halicin is different from known antibiotics. Will it overcome existing antibiotic resistance genes, right? That makes sense. So we took just wild type E. coli, and then we transformed it with plasmids carrying a bunch of different antibiotic resistance genes, right? And then we just asked, does the minimum inhibitory concentration, right, the minimum concentration of halicin necessary to inhibit growth, does the MIC change plus or minus these different antibiotic resistance genes? And we can see it stays largely the same. So it was able to overcome existing antibiotic resistance genes. And then one of the last, in vitro experiments that I thought was super cool and wanted to talk about was this one. So there's this there are two main bacterial survival strategies, right? One of them is antibiotic resistance, which we all know and love, right? And then the other one is antibiotic tolerance. And tolerance
Yeah.
isn't as well studied, but tolerance is the case where you have a genetically antibiotic susceptible bacterium that fails to be eradicated by a conventionally bactericidal antibiotic, right? And there's a bunch of contention. Nobody really knows why, but a common feature seems to be metabolic repression, right? Cells that fail to be eradicated by bactericidal drugs tend to have some sort of metabolic repression. But I was interested, does halicin potentially have the ability to eradicate such metabolically repressed antibiotic tolerant populations of E. coli? So right here, we're looking at a plot. So this is time on the horizontal axis, and this is viability on the vertical axis. In dark blue here, this is just E. coli growing in a tube, right? Nothing fancy, unperturbed. In light blue here, we have E. coli treated with 10 times the MIC of the antibiotic ampicillin, which is bactericidal. And we can see if we take E. coli and treat it with ampicillin, a bunch of cells die, but then they stop dying, and you have this population of cells that fails to be eradicated, right? These are your antibiotic tolerant cells. So what we did to see if halicin had activity against this population is we took E. coli, treated with ampicillin, got our antibiotic tolerant population, and then we treated with increasing concentrations of halicin, right? And we observed that halicin was able to eradicate these antibiotic tolerant populations as a function of concentration. So for me, this was like super cool. And that's when we got really excited about this molecule and then started investigating mechanism and all this stuff, right? But it was like this collection of experiments that kind of painted the picture of the potential capabilities of this compound, right?
Yeah.
And like you said, I mean, there's a million experiments in that paper, so people
Right.
should
So
read it.
unless you're working with mice or know more about mechanism, then definitely go check that out.
Yeah.